Good morning, everyone, and welcome to Global Compliance Panel's live webinar on statistical procedures needed for compliance with the guidance on process validation. My name is Michael, and I'll be your host today. On behalf of the Global Compliance Panel team, I'd like to thank you all for being part of this event. Today's webinar is being presented by Mr. Stephen S. Kuara. Mr. Kuara, a PhD, is the founder and the principal of GXP Biotechnology LLC, a consulting firm that works in the areas covered by the GLP and the GMP of drugs, biologics, and nutraceuticals. Steve has over 30 years of experience in supervising quality control laboratories, including an animal testing facility, and in performing GLP and GMP audits of internal and external testing laboratories. Steve has participated in the development of drugs and biologicals through all phases of clinical research and final product production. We are honored to have Dr. Kuara with us to present today's webinar. And before we begin, ladies and gentlemen, I just want to quickly outline today's program. This webinar is for a 90-minute duration. First, Dr. Kuara will take you through today's webinar, and he's going to highlight the areas that would be covered, and then he would share with you his presentation. And I just want to keep you all informed that once part of this teleconference, you all have been placed on mute and would remain so till the Q&A begins, which is ideally towards the end of the session. Again, this is to ensure that there is no discontinuity and to ensure that the presenter could speak clearly so that most of you could benefit from this webinar. And also, we all request you to hold your questions back, like I said, till the Q&A window begins. 10 minutes of time will be allotted to you during the Q&A session. And for any reason, ladies and gentlemen, if you do get logged out of this training session, just follow the same procedure to join in again. Now that we're all ready to start, I do request Dr. Kuara to take it from here. Dr. Kuara?
Thank you very much, Michael. We are going to be covering the guidance document on process validation. Now, I will warn you, the document that I am using for today's presentation came out in January of 2011, and we are on the first revision of that document. Now, there are some other process validation documents that are out there. There's the draft guidance that came out in 2008. And then there was the initial process validation guidance, and like I say, we're currently dealing with revision 1. Do not, don't get confused with the different guidance documents. Now, I will warn you, there are some slight differences among them, especially between the draft guidance and the current guidance. The general conclusions in terms of what kind of statistical procedures you need are the same. The differences have to do with some, minor, I wouldn't call them minor, but some procedural issues that were removed from the draft guidance and showed up and do not appear in the final guidance. Now, what we're going to be talking about today is this guidance document, and basically what I intend to do is to go through it more or less in series. Now, one of the things I want to warn you about right now, especially for those of you who are non-statisticians, is don't run to a software package. I have found that there's a problem here is that a lot of people go to a software package and they don't really understand what's going on in the software, mainly because they didn't study the statistics that are required. And as a result, they treat these software packages as though they're magical things. I mean, you enter the data in one column and in a certain order, and you press a button or an icon, and wow, look, these answers come out. Now, one of the problems is that people, in many cases, don't understand what they have just done. And this is why I say don't run to the software package until you have received training or you understand what is going to happen in the software package. Now, I will also warn you that there are some statistical software packages out there where the designers of the package, well, actually, it'll be the sales engineer that you talk to, are very reluctant for some reason to tell you what the different actions are based upon and what their command structure looks like in the software package. And I will advise you right now, when you have these people who won't talk to you and won't describe things of how the package works, I suggest it's time to move on and go seek another one. There are a couple of packages that I'm aware of that are very good, and their sales engineers are more than willing to talk to you. Now, question is, what does it cover? The problem with this document, if we go back here, notice that it was issued by CEDAR, CBER, and interestingly enough, the Center for Veterinary Medicine is also included. But notice that there are a couple of centers, FDA centers, that aren't included. And so let's talk about what is and isn't included in this package. Now, initially, it's basically human and animal drugs and biological products, and it includes the APIs. Now, notice that if you go to Q7, the guidance document on APIs, on GMPs for APIs, it does tell you that you should do a process validation. So your API manufacturers still have to do process validations on their processes, and this guidance document does cover what they need to do in those cases. Now, the guidance puts everything into a product life cycle concept. And let me see something here. Something just dawned on me. Okay, I've got a thing about what it does cover. Okay, let me back up. I got out of sequence here. Now, the time duration. The old system of doing process validation was that you did 3 demonstration lots for your NDA, and you did them and submitted them, and pretty much that was it. And you didn't do it again. You could make product for 30 years, and you did not go back and revalidate the process. Except, of course, if FDA told you that you needed to. Now, what we're dealing with now is the life cycle concept. And the life cycle concept means that you start with process development, you qualify the commercial process, and then you maintain the process during routine production. Now, let me warn you. This means that you continue to do process validation, verification, whatever you want to call it. And this goes on through the lifetime of the process and that you're using. Now, the life cycle, remember, it starts with product development and it ends when you discontinue the product. Now, one other thing here. Notice that I have a statement here that's in italics, as opposed to regular print. When you see something in italics, this is my statement, as opposed to things that are in normal print. Normal print are statements that were taken directly from the guidance document. The things in italics are things that I have injected into the discussion. Now, okay, let's get back to what kind of products are covered by the guidance. You do human drugs, veterinary drugs, biologicals, and biotechnology products. Now, remember, biotechnology products include the biologics that are covered by CBER, as well as the biologics covered by CDER. And biotechnology products also include cell therapy and related products. Now, finished products and APIs are covered, as we talked about earlier, and also the drug part of a combination product. You know, a drug and medical device product, for instance, say a infusion pump that comes with a preloaded cartridge. The drug that's in the cartridge is covered by this guidance document, or the biologic that would be included in this thing is covered. The things that are not covered are things like, well, type A medicated articles and medicated feeds, and well, I didn't put it in italics, but basically I've injected here, these are veterinary products. And some of these medicated feeds are the kinds of feeds that are used in feedlots when they're fattening cattle and pigs. Now, medical devices are not covered, and dietary supplements are not covered. Now, dietary supplements are classified as food, so you have to look to the food GMPs to look at their process validations. The medical devices, and then human tissues intended for transplantation. These two classes, devices and transplantable tissues, have their own guidance documents. So, you know, if you're working in these areas, you need to go and look at those guidance documents, not this one. Now, this is a definition of process validation as given in the document. And remember that we've reached the point now when FDA says scientific evidence, it usually means that they want to see statistical data and statistical tests used. And, you know, it has sort of evolved because in recent years, FDA has become much more insistent upon seeing statistical studies done in the manufacturing area as well as in the clinical trial area. Now, automated processes. There's a statement here that the guidance doesn't specifically address the validation of automated systems. However, it's relevant to those validations. And what they don't exactly point out here is that automated equipment and processing comes under Part 11, 21 CFR Part 11. So the main part of your validation would come under Part 11, but this guidance document does apply to those validations as well, especially software validations. Now, this is a statement from FDA that process validation contributes significantly to drug quality, and they talk about the basic principles. And they then tell you that this principle incorporates the understanding that the following conditions exist. So they're finally telling you what they believe you should be doing. And these are the three things. And the whole point here is that, you know, you can't do just testing alone to assure quality. And part of the problem here, remember, is that you cannot test everything. You know, it's one of these things called test to destruction, where you do so much testing that you destroy the product you're trying to make. And good example of this, for instance, would be sterility testing, where you have to inoculate your product into test medium, and that kind of destroys the product right there. And so the thing is that you have to depend upon statistical procedures to say that if you sample a certain part of your product, that the results you get for that particular part of the product applies to the whole lot or batch that you have made. Now, they also talk about each step being controlled, and we'll talk some more about exactly what a controlled manufacturing process is from their point of view. But here's the thing. There are three parts to a process validation. The initial one occurs at the design stage, and it requires a definition of the process during this stage. And this definition usually requires statistical studies to basically define which parts of the manufacturing process are important and which ones are not in terms of introducing variation into the system. And then in the process qualification stage, this is what used to be called process validation. It's the stage 2 here. The idea is to prove that the process is reproducible. Now, you know, this leads to all sorts of things, including the old requirement that you had to have 3 consecutive lots. You know, if you can't make 3 consecutive lots that all meet spec, then your system is considered to be unstable. And then finally, your continued process verification, not validation, verification. And you're looking for proof that your process is in a state of control. And I've highlighted state of control here because that statement, that phrase comes up in a lot of documents. Now, the elements of the stages. Defining the process usually requires definition of the design space. And, okay, all of these lowercase things here are about smaller fonts. These are my injections into the discussion. Now, during the process design, and this would include process development, really, during this process, you need to define the design space. And the design space is basically the region, and it's, you know, multidimensional, where you can operate and still make the same product. And it allows you to, well, FDA in another guidance document allows you to make changes within the design space without having to report it as a major change. Now, the thing here is that defining the design space requires a definition of what is called the CQAs, the critical to quality attributes. And this requires the application of design of experiment methods, DOE methods. And we will say more about DOE as we go. The term reproducible requires an understanding of the variance of the different steps. And this is done fairly simply through calculation of the variance over multiple runs. And then state of control. The state of control requires the application of SPC, or statistical process control, to monitor the process. Now, process validation has to start at the development stage, and this is their statement. You know, you start with information and knowledge from product and process development. This means that your development people have to document their activities very carefully and make that information available to the people who end up running the final process. And part of this understanding and knowledge is your basis for specs, for setting, the process parameters to produce your quality attributes. Manufacturers should understand it. There's a whole series here about variation. This is one of FDA's biggest bugaboos. When you submit your NDA, you have a process described in it. And FDA expects you to make a product that follows that process as closely as possible. And they are looking here to see that, well, you're allowed a certain amount of variation. I mean, this is normal for any process. But they want to see that you can control the system within the variation that you specify. Now, this variation, you see it in a lot of things as things like the standard deviation or the variance of a procedure. And this is nothing more than the plain old beginner statistics that you were taught when you were in school. At least I hope you were taught these things. You calculate things, you run your process, and you measure your different parameters and output at different stages, and you look at the variation in those stages. And this allows you to understand where are you getting variation in your process, how do you detect the variation, what's the impact of the variation in the process. And this part here usually shows up in studies that are known as the analysis of variance, ANOVA studies, or design of experiments. And then once you've defined where the variation comes from and what the impact is, then you take methods to control the variation. And this requires statistical studies of the outcome of runs, but it also includes the idea that you will have the manufacturing knowledge to control these steps. You're expected to understand the process. And there's a statement here that focusing exclusively on qualification efforts without understanding the process may not lead to adequate assurance of quality. What FDA is trying to get at here is that there are some companies where people focus entirely upon doing these 3 qualification lots, and they don't care about things afterward. They just want to throw out 3 lots that look good, and they send that to FDA, and they really don't understand what's going on in the process. The thing is that if you're really careful, and especially those of you who have so-called validation groups, the validation group can make 3 lots. They're concentrated on those 3 lots. And they make the 3 lots without really understanding where the sources of variations are. One of the biggest eye-openers I had in my career was one day when I went over to the process engineering department, and I asked them, I said, can you tell me at what steps in the process you expect to see the largest amount of variation? And we were developing a new process at this point. And people just sort of sat there with their mouths open, staring at me, and that's when I realized we were in real trouble. The thing is that in those days, that was when I was the head of a QC department, and I was trying to determine where we should concentrate our testing efforts. Which samples, when we should take samples, at what stage in the process, how many replicates we needed in samples, and all this. And to do that, I needed to know where the variations were coming from. And my process engineers didn't seem to have an answer. They didn't seem to know how to answer that question. And I realized that at that time that we had this validation group that was doing nothing more than trying to make 3 lots in a row that met specs. And once they did that, they just sort of walked away from the process. And there was no effort made, especially at the development level, to understand where the variations could come in. There was no risk analysis to say that this one particular step was very important and all that. Now, to do this continuous process verification that's going to be talked about as stage 3 here, you need 2 things. You need change control, which is really sort of a documentation issue, but it's an issue all through the company. It's a management issue. And then the part that the statistician will give you is called statistical process control. It's the SPC. And, there's a whole bunch of acronyms and abbreviations that have come up. There's the abbreviation CPV that refers to continuous process verification. It's used instead of saying stage 3. So anyway, we'll talk more about that. Now, I have here a section on comments that anytime you see comments, these are my comments. They're not FDA's comments. The understanding and controlling variation, you need to be able to calculate variances and standard deviations. For a lot of you, you've got pocket calculators that can do this. You don't really need to run to a main computer or anything to do this. It's fairly easy to do. Believe it or not, you can actually do it with pencil and paper. You have to be able to take square roots. That's the only thing. Now, the calculations of confidence intervals, and let me kill this thing. There's an argument that's going on right now for you statisticians out there as to whether or not you should set your specs using confidence intervals or tolerance intervals. Personally, I come down on the side of tolerance intervals. But again, you have to remember when you use tolerance or confidence intervals that you have to understand what's going on, because I have seen a lot of places that have set specs that are unnecessarily tight because they have used a confidence interval based on too many replicates. And that's a big question. Now, when you come down to the question of, okay, you've got these variance calculations, and now you want to know if 2 variances are equal or different. And the procedure to do this is to use what is called an F-test. And in this particular case, it's a one-sided F-test. But the whole idea here is before you even go into this, you can compare the variations that you get in your system by doing a so-called ANOVA, an analysis of variance. This allows you to understand which of your processes or which steps are giving you the most variation. And then you can also use what are called screening studies or screening design of experiment studies to determine the importance of various steps in terms of inserting variation into your system. Now, your screening DOE, there are some very large scale studies that you can do. And my advice to you is start simple. Don't go into a huge study, 11 factors and all this sort of stuff, because all you're going to do is drive yourself crazy during the initial study. Start off simple. When people get used to working with the simple DOE studies, then you can possibly expand it to larger scale studies. Now, the process in the NDA needs to be reproducible. And your calculated intervals are going to show the amount of variation that might be expected by both you and FDA. So make sure you're doing a good job in calculating these intervals. Now, okay, these are the only formulas I'm going to show you. These are different formulas for calculating the variance. Now, the middle formula here, this is actually sort of the defining form for defining the variance. You take a bunch of numbers, supposedly that are measuring the same thing. And remember this, you have to be able to say that I am measuring the same thing. And if the world were perfect and there were no variations, all these numbers will come out to be identical. Now, you and I know that in real life, in real work, the numbers do not come out to be identical. There will be variation. And this is a normal thing. This is because the world is not a perfect place. There is wobble in the system. So you get a bunch of numbers, and you take the average of those numbers. That's what this X bar here is saying. You take the average of these numbers, and you subtract it from the individual observations. Okay, X sub I. This could be 1, 2, 3, 4, whatever. And each time you make this subtraction, you square it, and then you sum it. That's what the sigma is. And it's divided by n minus 1. Now, a couple of comments here. You square this thing because of the fact that if you look at this, you will realize that depending upon x, what x is here, x sub I, you will get either a positive or a negative number. Now remember, if everything is perfect, right, these x sub i's should be symmetrically distributed around the average. There should be larger numbers and smaller numbers. And if you look at this whole thing, if you have a huge number of x's, what you will find is that, you see, these things are called the deviations. And the difference here, the sum of the differences here will go to 0, right, if n is large, because you will have so many variations. Eventually, you should end up with an equal number on both sides. So it'll appear to be 0. But the problem is that we're not interested in whether the difference was positive or negative. We're mainly concerned that there was a difference, high or low. So by squaring the number here, the negative numbers become positive, and you can sum all of them as positive numbers. The other thing that I want you to remember is you divide not by n, which is the number of x's here that you threw into this average. This n is this is what is called the degrees of freedom. It's n minus 1. Because in making this average here, you had to total or sum the numbers. And that took away 1 degree of freedom from your n's. So it's n minus 1. And remember this because there are what I call population statistics. In population or large number statistics, you use n. You divide by n itself. Now, let's face it. You get into this thing of, okay, whether to use n or n minus 1. Well, if you have large numbers, say you have 100 replicates here. You have x sub I goes up to x sub 100. It doesn't make too much of a difference whether you divide by 99 or 100. Okay, let's face it, the answer is going to come out pretty much the same. However, if you're dealing with small number statistics, which we're all trying to do because we don't want to run too many replicates or too many experiments, too many samples, all this stuff, let's face it, according to some bosses, even 3 replicates is too many. It does make a difference. If you divide by 3 versus dividing by 2, or 3 versus 4, the answer comes out to be very different at that level. And so it is important to remember that you're dealing with n minus 1 here. I will warn you that if you're using Excel to do your calculations, many people have had problems with Excel because Excel has 2 standard deviation functions in it. In one of the standard deviation functions, you divide by n minus 1, and in the other one, you divide by n itself. And when you're doing small number statistics, like I said, there will be a big difference whether you're using n or n minus 1. And a lot of people don't know the difference, and they don't take the trouble to go into Excel and figure out which formula is being used to calculate the 2 different standard deviations. So go and take a look at the standard deviation you calculated with Excel. Now remember, the standard deviation is nothing more than the square root of the variance here. Now, the variance is useful because this is a positive number, and you can manipulate it like any other number. You can add them together, you can subtract them, and all this. The standard deviation, however, is a plus-minus number. It's plus or minus the standard deviation. And what happens is that you cannot simply add together 2 standard deviations, because 2 standard deviations added to each other you have the result of the plus-plus addition, the plus-minus addition, the minus-plus addition, and the minus-minus addition. So theoretically, you have 4 different answers here, which, drives everybody crazy. So if you work with the variance, it's a lot better. The reason we use standard deviations is that when you do a variance calculation, and presumably you're attaching units to your measurements the way you're supposed to and the way I hope they taught you to do in school, what you end up with is a unit squared, which may or may not make sense. For instance, if you're doing concentrations and you have micromoles up here, what is a squared micromole, or for that matter, a squared milligram? You don't know. It has no meaning when it's as the variance. So in order to deal with this thing in the real world, we take the square root and we deal with the standard deviation, which does have units in the normal sense. You would have milligrams, you would have milliliters. But you don't get this when you're dealing with the square. So this is one of the reasons why we work with standard deviations in a lot of cases. But remember that in the statistics, it's a lot easier to work with the variance. And the variance makes more sense on a statistical level. Now, the reason I have three equations here, this equation, all three of these equations are equivalent. You can take this middle equation here and massage it around to form this equation on the top here, or the equation on the bottom here. Now, the thing is, you find that this first equation on the top comes out and it appears in many statistical calculations. So actually, you're talking about the variance here, and you find that this appears in many other discussions, especially in things like linear regression and also in design of experiments. This one down here, for some reason that I haven't quite understood, is very popular among the people who do calculator problems. Apparently, it's easier to program this equation than it is to program this one. So you will find that in many calculator or computer programs, actually plug in this equation here. In any case, the answer is the same. They're all variances. Now, the comments on process control. First of all, remember that GMP seeks a state of control. That's one of the whole reasons behind the GMP rules, is that you want a process that operates in a state of control. You have to have change control systems, you have to have SPC, and for continuous process verification. Now remember this, some people try to substitute these things, but the continuous process verification is something you do all through the year, not just once a year as with the annual product review. The annual product review is part of your continuous process verification. It does not replace the continuous process verification. And for those of you who work with this sort of thing, notice that if you've got only a single process, it used to be that, well, okay, at the end of the year or at the beginning of the year, you just assign some people to go and take a look at the process and do the annual product review. You can't do that anymore. You have to have one or two people who are looking at the process all the time and monitoring the whole thing. Now, during your process development stage, you need to do what are called process capability calculations. And these are called capability indices. You see them as C sub P or C sub PK or P sub PK, depending upon what notation system your statistician uses. Actually, you don't need a statistician for this. I mean, any engineer or technician should be able to make these calculations. Now, the capability indices are kind of interesting because they were originally set, the target was originally set to be plus or minus 3 sigma around the process average. But modern manufacturing, and I would say this is since the 1980s, people have tried to target a plus or minus 6 sigma environment, not a plus or minus 3 sigma. But to get to 6 sigma, you need to have very good control, and you have to have a very stable process. So this is something that you do want to look at in your work. Legacy products. Some of you have got legacy products and you're saying, hey, what am I going to do with this? We didn't do a process validation when we introduced it. We don't have any development data anymore, or the development data are very weird, and we're not too sure exactly what they were thinking about when they did the study. All of this. Now, in the guidance document, it just says basically you can use the knowledge gained from your original process development, which some of you can't even find because you never really did any decent process development. You just copied the process. And so what it is that you use your manufacturing experience, look back over previous lots and see what kind of data you can get out of it, especially in terms of variation. If you look at your past lots, you should be able to get a good idea of where you're seeing a lot of glitches and variations in your process. And based on that, you can introduce numbers into statistical calculations. Now, basically what they're saying here is for legacy products, you should start with the so-called stage 3 activities. In other words, do a continuous process verification right now. And that will get you started. And then as you're going along, you can generate some of the additional information about how you justify your process control or critical process parameters. Now, here we go into the process validations. And notice this. Let me back up here. You see this thing up here? These are indications from the guidance document. So this is section 2, subject B, paragraph 3. Or, well, the 3 I've sort of injected myself. But we are now into section 3 of the guidance document. And this is the title. The A I've just sort of stuck on. This is the first half of what I'm going to talk about, first part anyway. But basically it tells you the GMP regulations regarding sampling put forth a requirements for validation. And remember, samples must represent the batch under analysis. Sampling plan must result now in statistical confidence, and batch must meet predetermined specifications. Now, remember, these statements up here say that your sampling plan has to be statistically justified. You must know the level of risk that your company is willing to accept, a risk of making a so-called type 1 error, where you find a difference due to statistical variation alone. And also that the specifications you set here should have been set with the variance in mind, and which would require, at least a tolerance interval calculation. And this sort of thing, statistically defined number of sample replicates at a defined level of risk. And, one of the things you find a lot of times is that your management doesn't know what the risk is or what risk they're willing to accept. There's always this naive thing of, no, we don't want any risk at all. Well, not getting any risk at all means that you have to go to a huge number of replicates. And, I don't think you're willing to accept the costs that are involved with having 100% confidence or no risk at all. Part B here, second principle requires that in-process specs are derived from previously acceptable process average. What this is telling you here is that when you go into manufacturing, there is always the question of, well, what do we use to set the specs? Well, Basically, you should have specs set by looking at your development process. You know, your development people should have given you data on the process and should have done the initial uh, setting of the specifications based on the variations that they saw. And so these should be statistically justified. You know, determine application of suitable statistical procedures. And you know, you need to look at process performance and your batch-to-batch variability. Now remember, with legacy products, you should be able to do this by doing a look back or a retrospective study on what your process is giving you. And based on this, you should be able to either justify your specs or not justify them. Now, if you've been doing annual product reviews through time, you should have the data, and in fact, you should have been adjusting your specs over time based on your annual product reviews. And now you would do nothing more than collect these reviews together and use this as your basis for justifying the existing specifications. Now remember, previous experience. So at every stage in the line, you start using specifications based on what the previous people found. And you know, even in development, you know what was found in R&D, what was found during the early stages of the work, bench scale, things like that, and you use that as your, you know, that's your starting specs. Now, FDA has said repeatedly that they realize that specifications will change over time, and that, you know, it'll be necessary to adjust specs and make them evolve. Now remember, when you do this, you need to have your statistical variations in mind, and you need to be able to talk about them. But it's perfectly acceptable to start with wide specs and then narrow them later as you got more data and experience. And let me warn you, I have seen several companies where FDA has come and asked them, basically made the statement, look, you know, we know that your process operates at this point with a plus or minus 8%, and yet you have your specs set at plus or minus 50%. This is not acceptable. We expect you to tighten your specifications. And so you need to go back in and look at these things, because sooner or later, during a review or an inspection, you will have a person that'll make this statement, and you're going to end up doing it anyway. So it's much better if you can do the adjustment yourself before it becomes an issue with FDA. Section 4 here, recommendations. Now, these are recommendations from FDA, okay? We recommend, this is FDA saying this. Now, they want to see an integrated team approach to process validation. So what are they looking at? They want people with expertise from a variety of disciplines: process engineering, industrial pharmacy, analytical chemistry, micro statistics, manufacturing, and quality assurance. They expect to see all of these people involved in your process validation. Now, this does not say that these are all different individuals. You know, your industrial pharmacy person could also be a process engineer with expertise in pharmaceutical engineering. There is a society, international society of pharmaceutical engineering. And your microbiologist, you know, one of the problems here, of course, is that you want the microbiologist to be experienced in clean room operations, in things like objectionable organisms, culture of that. And your statistician, and let me warn you, your statistician who handles your clinical work, your clinical studies, may not be satisfactory for handling your manufacturing statistics. Statisticians have specialties, and they work in different areas. They're just like doctors or chemists or any other professional. They have their specialties, and you will find that the clinical study specialists do not necessarily know the statistics that you need for doing manufacturing studies. Now, you have to have project plans for your process validation, and you have to have full support of senior management. You know, this is one of the biggest things, is lack of support from senior management. In your process design, this is stage one. Remember, there's 3 different stages to a process validation. Okay, stage one is process design. And during your process design, you need design of experiment studies, DOE. Now, exactly what is DOE? If you're not familiar with the term, DOE refers to what are known as factorial studies. These are studies where you do many different factors or elements of your process, and you look at the outcome that happens as you make adjustments. And usually these are so-called two-level studies where you do high, low, plus, minus, etc., etc. These are factorial studies, and it's, of course, if you've only taken low-level statistics, this can sort of frighten you, but it's not really frightening. Design of experiments is just as easy to do as anything else, except that, yeah, you do have to know some algebra. I'm not going to talk very much about it in the sense that, you're looking at a 1-week course to do design of experiments for manufacturing. But, just get the idea. It's fractional, factorial, or factorial experiments. Now, the problem with factorial experiments is that the numbers get large. So a lot of times people do what are called fractional factorials. You do pay a price for that, but you need to do it, and this should be done really at the development stage of your work, because in this stage, you want to define the relationships between your critical process parameters and your critical quality attributes. During design, your design people should identify what the critical quality attributes are, and at that point, determine also what are the critical process parameters that give you the critical quality attributes. And this can be done through design of experiment studies, where you can show relationships between them, or non-relationships if you want to do that. Process design, again, you need to do risk analysis. If you're going to you do risk analysis, and a lot of times you'll end up with these arguments about, well, this element is important, this one is not, and somebody at a meeting insists that something is not, while somebody insists that it is, etc., etc. Well, design of experiment studies will allow you to look at these parameters and decide which ones really are important and which ones are not. In other words, which ones can affect the outcome of your critical quality attributes. And from that, you can provide justification for establishing ranges. Incoming component quality. This is a major issue with FDA because it's been found over the years that a large percentage, and I'm talking about like 20% of product failures are due to bad raw material or incoming component quality. And you really need to control this. But again, the question is which ones need to be controlled and which ones are important. Now, notice this last statement. FDA does not generally expect manufacturers to develop and test the process until it fails. Okay, you don't need to push it to the point of failure. They assume that you're not going to be operating at the limits of failure, that you will have a process where you can work within parameters that avoid the possibility of massive failures. At least I hope you are. Now, design of experiment studies. These are multifactorial experiments, and you're looking at the factors that can affect the process. And basically what you're looking at are the process parameters. And that's your input. Your output is going to be your critical to quality attributes. And what you do is you make high, low, or plus minus variations in your critical process parameters and see how that affects your critical to quality attributes. And the interesting thing is that you're going to find out that there are many things that you thought were critical quality attributes that are not affected by critical process parameters. And you can eliminate these things. If they don't have a big effect, then you don't need to monitor them as closely as the ones that do have an effect. So this allows you to put your effort into the right area. Now, the problem here is that, people start a lot of times, especially the inexperienced or, people, they start with what are called best guesses. And so you have your so-called one factor at a time approach. This is where you've got, 6 different factors that you're at a particular step, and you hold 5 of them constant, and you vary the 6th one. And this is very inefficient. And we can, it's easy to show that this results in more work than a design experiment approach. When you look at DOE methods, sometimes you hear them they're called randomized blocks, Latin square designs, factorial designs. Often you get an exponentially increasing number of experiments, and this is what scares management. Now, one of the reasons you get this thing is that, you see, if you do DOE studies, you can calculate in advance how many studies you're going to need. And, and this looks large a lot of times, presented up front. Now, the problem is that the people who do OFAT methods, one factor at a time, a lot of times they'll look at it and say, oh, no, we don't need 8 more studies. All I need is 5 more. And generally what happens is that after they've done the 5, it becomes, oh, well, I guess we need to do a couple more. And then they do the couple more, and then they say, oh, well, we need to do 4 more. And so on and so forth. And eventually what you will end up with is more experiments than you will have to do with a DOE approach. The trouble is the DOE approach showed you the large number to begin with, which scared people. And the OFAT people come with low numbers, and they lowball the system, in other words. The problem here is that it has been demonstrated, and well, it can be calculated actually, that for the same amount of information, an OFAT procedure will always require more studies than a DOE study. So remember that designed experiments minimize the number of studies you need to do. And then you can do things like using fractional factorial designs, or you can apply screening methods. The two most popular screening methods around are Plackett-Berman studies or Taguchi studies. And I will tell you that as far as I'm concerned, these two procedures are more or less equivalent. And, you know, which set you use kind of depends upon your previous training and personal inclination and all this. So, you know, if a statistician wants to use one or the other, that's fine, as long as they use one of these. Process qualification step. Now, this is the part that normally or used to be known as process validation. Now, there's two elements to this. One is the facility, equipment, and utilities, and second is the process performance qualification, or the PPQ. Now, here's the thing. At stage 2, you need to be following GMP compliant methods. Remember, stage 1 was development. And in development, you don't need to strictly follow GMP procedures. However, you know, you need to have reproducible methods. You need to document everything and all this. So pretty much you're following GMP, but you don't need to be strict about it. At stage 2 of your process validation, you do need to go into full GMPs. And this is the thing. See, you have to complete stage 2 before you can release product. And this is kind of misleading because normally you can't release product. But there are circumstances where you would be allowed to release product after completing a stage 2. So, you know, at stage 2, you have to go into full GMPs. Now, here's your process performance qualification, your PPQ. You use your cumulative data, and that includes your designed experiments that you did in development, and your laboratory results, your pilot studies, your lab scale results. All of this are used to establish the PPQ. And, you know, FDA says we strongly recommend. And, you know, that's basically telling you you better do this or have a good explanation for why you're not. That you employ objective measures, and they say, for instance, statistical metrics. This, you know, some of these statistical metrics are not very scary. I mean, it's nothing more than averages, standard deviations, and those kinds of tolerance intervals, and those types of statistics. Now, it says wherever feasible and meaningful, and if you can argue that they're not feasible or not meaningful, then you don't need to do it. Now, notice this. They expect the PPQ to have a higher level of sampling, additional testing, and greater scrutiny. Now, a higher level of sampling gives you a higher level of confidence, and that's basically what they're looking at. And one of the things that you need to look at here is to tell your statistician that perhaps you want to run your PPQ studies at a 99% confidence level or 99% level, even though you may know that in your actual manufacturing or your commercial runs, you're only going to do 95%. And, you know, But at this level, and when you're doing the PPQ, you want that higher level of sampling. And you need to write a protocol, and the protocol should discuss things like the number of samples. And you see, this is the thing, at sufficient statistical confidence. You know, are you willing to accept only 90%? Well, probably not in the days of Six Sigma. You know, you probably want something more like 99% at this level. And both within a batch and between batches, and the confidence level should be based on risk analysis. In other words, if you have a step where the risk analysis shows that the consequences of variation in this step are not very huge in terms of the quality of the product, then, you know, maybe you don't need to have a high level of confidence for the results at that stage. So you might only do enough samples, for instance, to give you 90% confidence. Whereas if you have a step, for instance, a step that generates an impurity that's known to be toxic, you probably want to have about a 99% level of confidence in the results you get at that step. So you would need to adjust your level of confidence in your statistical studies. Now, stage 3, continued process verification. This is where basically what we're looking at here is statistical process control. And what you're doing here is you've basically validated your process at stage 2. And what you're doing at stage 3 is providing data and evidence that says that your process remains in a state of control. And to do this, you're looking at SPC, statistical process control. Now, they're telling you right up front that they have a regulation that tells you that you should be collecting and analyzing the product and process data. And that's 180E. Actually, this is the regulation that establishes the need to do an annual product review. But the problem here, of course, is that they're telling you you should be collecting this stuff over a continuous period of time, not just once a year. And then the thing is, data should be statistically trended and reviewed by trained personnel. Now, again, trained personnel doesn't mean just your statistician. If you're a QC person, or, well, some of the QA people are technically qualified. If you're technically qualified, you should be able to look at this stuff and understand what your trending studies are showing you. And again, you know, you should verify quality attributes are being appropriately controlled now throughout the process. So you can't just forget about this stuff, okay? They recommend that a statistician or a person with adequate training in SPC develop the whole thing. Now, I will tell you, you don't need to be a statistician to do SPC. And, well, you know, I'm not a statistician, yet I did it for years. And basically, the training is available out there. And, you know, like I say, go through the training before you use a software package. There's too many companies where people have a software package, and that package automatically picks up data from the LIMS system and inserts it into a continuous chart that monitors the process as an SPC chart. The trouble is that when you do this, people don't look at the result. It's amazing how many times during an OOS investigation, for instance, you go in and you look at SPC charts and you realize that the problem began in some cases months before the actual out-of-spec result was obtained. And what happened there was that nobody was looking at it. People that did look at it said, it's a little high, but it's still, well, it's in spec. I'm not going to worry about it. Things like this. And then finally, when they do get to the out-of-spec test result, then we've got a major problem on our hands. And this is one of the big problems with software packages, is that not only do people treat them as though they're magic, but they don't even listen to their own magic. The process starts showing that, things are going out of trend. You should be looking at your charts at that point, but they're ignored. And everything's routine, no major effects needed or actions needed. And there's a real problem here. So make sure, and for those of you who are supervisors and managers, make sure that these things are reviewed minimum of once a week. And it depends upon how many lots you're making. If you're making 4 lots a day, let's face it, once a week is not enough. So, and it might need to be every 2 days or even daily. Now, you're looking at process stability and capability from your production data. So again, you should still be calculating your capability indices. But if your SPC charts are stable, you probably don't need to look at capability indices. And finally, it says here, the quality unit should review the information. So even if manufacturing is collecting the information, it should be sent on to the quality unit for review as well. So again, the old GMP principle of 2 sets of eyes. And hopefully the quality unit will have a statistician or somebody trained in statistics who will be able to look at the data. You have FDA saying there's many tools and techniques, some statistical and others more qualitative. And, basically they're saying things like, well, detect variation. This is, of course, done through your SPC charts. But once you detect the variation, you have to characterize it and determine root cause. So we go into root cause analysis as well. They recommend that you use quantitative statistical methods, again, wherever appropriate and feasible. You want to look at intra-batch as well as inter-batch variation. Now remember that you're looking at variation within lots as well as from lot to lot. So both sets need to be monitored. And let's face it, you cannot see intra-batch or within batch variation unless you take more than one sample. And, question is, how many samples should be taken? A statistician can calculate for the level of risk you're willing to accept how many replicate samples you should be taking. And this can be done, I'm only too glad to teach a course on it, but you can do this. Finally, they want continued monitoring and sampling of process parameters, quality attributes, and again, generate significant variability estimates. Make sure your monitoring is adjusted to a statistically appropriate and representative level. Again, if your statistics are showing you that there's very little variation induced due to a step, then, your monitoring of that step does not need to be as rigorous. And it's the steps in the process that give you the variation. Those are the ones that you really need to look at. You need to look at it with a sufficient number of replicates. And, you adjust your work based on the variation you see in your process. And this is something that's fairly easy to monitor and keep track of. So I encourage you, get the statistical training or get a statistician. I will warn you that nowadays hiring a person with sufficient statistical training, especially in manufacturing, is not that easy. So sometimes, and it's easier sometimes to just go out and get the training. The state of control. Now, a process that is operating in a state of control, it's reproducible and stable. And this is the whole thing. You want to get to this state of control in your manufacturing process. Because if you have this, then you can predict future yields. Then you can make all kinds of predictions about future behavior. This allows you to say that, okay, next year we will have 50 good lots, and we should, in those 50 good lots, we should have the certain number of vials or product units or whatever you're doing, millions of units, whatever. And then this allows your company to do planning on how much it's, raw material it's going to need, how much worker time you're going to need, all this. And also in making business decisions. Where are we going with the company? Now, a system that operates in a state of control only shows what are called common cause variations and no assignable cause variations. Okay, this is the terminology that is used by people like Deming and Schuhart. Common cause variation is variation due to the process itself, and it's inherent in a given process. And you can't do much about it until you change the process. So it's usually said that common cause variations can only be fixed by management. It takes management activity in order to affect your process, your basic process variation. Now, assignable cause variation is usually a one-time thing, or, well, hopefully no more than once, but sometimes it happens over and over and over again. And it's due to something external to the process. Sometimes it's nothing more than a sleepy worker. Sometimes it's because of bad raw material. Sometimes it's because somebody made a mistake while adjusting the machine. All of these things, these are assignable cause variations. And these are things that actually can be fixed by the workers themselves. The workers themselves know that there's a problem here. And they can stop the process and tell you, what's wrong. Now, the whole idea behind process validation and all this work you're doing is to eliminate assignable cause variation. And then, if necessary, if the common cause variation is too large, for instance, if common cause is so large that it gives you, say 5% of a lot always fails, then it tells you you need to go into your process and work on the process to reduce these failures. But it's not a special thing. So this is the whole thing about getting to a state of control, is you minimize your losses and you minimize your problems because of variations in your system. Now, this is Deming's statement, and I will conclude with this. If your distribution theory, in other words, your control, if the normal distribution is not applicable because you lack control of your process, then anything any inference, statistical or otherwise. And you know, this includes things like planning for the future, how many lots you're going to make, how much product you're going to make, what your sales are going to be. All this is little better than conjecture. And you don't want this. And with that, I will wrap up this presentation and turn you back to Johnson for questions.
All right,
not
thank
Johnson,
you so much,
Michael.
All right, thank you, Steve. Ladies and gentlemen, like Steve just said, we'll go ahead with the question and answer session right now. So in the just concluded presentation, if you have any questions whatsoever, this would be the time. You could go ahead and click on the raise hand option on your participants panel, which is a palm-like icon, and you can directly put your question forward to Steve so that he could answer your question, or else you could go ahead and put your questions forward in the Q&A panel on your participant screen, and I will, or even Steve can access those questions and he can directly read them out and answer them to you. Or if you can't do either of the two, you could just send me a message on the chat messenger and I'll put the question forward to Steve. So if you have any questions, ladies and gentlemen, this would be the time.
Okay, one other comment here. If you don't ask a question and then an hour from now or tomorrow it dawns on you that you have a question about this presentation, please feel free to forward the question to Michael, and he will in turn get the question to me and I will respond to it. And I won't guarantee you an instant response, but I can tell you I will respond to it. So, you know, please feel free to forward questions and ask questions right now, in fact.
Absolutely. Like Steve just said, ladies and gentlemen, in case you come up with questions after the session as well, you could send me an email with your question and I'll put that forward to Steve and he'll definitely get back to you with an answer. And Steve, we in fact have a question on my chat messenger. I'll just put that question forward to you here, and it should be on your chat messenger right now. Just bear with me.
Okay.
And yep, there you go.
Okay. You haven't seen data integrity studies. Okay, test method validations, gauge. Okay, all of these studies, they're not required in the sense of you have to do it, but you should do them where appropriate. Now, let me give you a thing here. For instance, test method validations. You will see at the end of this document, if you read it well, not read it, but just go to the end of the document, there are comments made about test method validations. And basically what they're saying is that a test method is a process, and the process validation guidance document is applicable to test method validations. Now, the specifics are probably to be found in what is known as ICH Q2. But for instance, your test method validation, you do want to do test method validations. You do want to maintain the thing by doing SPC charts on your test methods and you know, maintaining them. The other thing, things like your gauge R&R, yes, you know, the equipment you use, all of that should have been qualified and checked. All of your equipment, your process machinery, this sort of thing. Now, this is one of the problems, see, is that because people charge into the process validation without realizing that certain things have to be in place before you do your process validation. And one of the things here is that you do your process validation. How are you monitoring it? Well, you need analytical methods. So those analytical methods should have been validated before you went into a process validation. Your equipment should have gone through equipment qualifications. You know, your DQ, IQ, OQ, PQ. Well, PQ maybe not so much, but you need to have qualified your equipment. You should have done gauge R&R studies where appropriate. All of these things, they're not required, but they should have been done in places where they are useful. And I guess that's my answer to the whole thing, is that there are all kinds of techniques, not just statistical techniques, that need to be executed in a process validation.
All right, thank you, Steve. Steve, I have a few more questions your way. The next one would be again on your chat screen right now.
Okay. Okay, I have, I have personally used the 95-99 system. There have been cases where I have used a 99-99, although I'll tell you that that gets to be kind of large at times. But yeah, the tolerance intervals, 95-99, pretty common. And, you know, for those of you who are not familiar with tolerance intervals, what we're talking about here is, for instance, we want 95% confidence that 99% of future results will fall within the interval, the tolerance interval. And that is how you set up your tolerance intervals. So you always come up with this question of how much confidence do you want and how much do you want to predict for the future. Now, for instance, the question also referred to a 99.73. You know, you can push that thing out. Well, theoretically you can push it to infinity, but then you consume the whole lot. Basically, you want to look at how much confidence do you want to have that your prediction will work, that the following results will fall within the interval. And that is kind of a management decision. Personally, I have used 95-99, like I said. But some people tried a 99-99, and you know, it kind of comes down to how much you can tolerate. I guess that's why it's called a tolerance interval. But
thank you, Steve. And Steve, the next question which I have here, I'll just read it out loud. It's on your chat screen, and at the same time, I'll just read it out. The question is, how do we measure intra and inter batches variability?
Okay, basically what you're looking at are lot-to-lot and within-lot variation. For instance, we have a lot of product, and you need to look at the units that this thing's made out of. And sometimes if you're just looking, for instance, in an API, you may not have units. You just into more or less a continuous processing thing. Basically, what you need to do is to take samples throughout the process. Now, everybody always says the most critical samples to take are like the first 2 units off the lot, off the filling line, and then take the last 2. You can take in between. Now, the number of units you take, this is where you get your confidence. You have to say, well, okay, I will do this, and I want to get an answer which will be an average and standard deviation. And the question that I need to know is, how much confidence can I have that my answer is close to the real, true situation? So based on that, you can calculate the number of replicates that you need. A statistician can do that for you. But okay, so you either take samples from within a batch, and you could do this, for instance, for 3 batches. It kind of depends upon the variation you see in the batches. But you look at this and you calculate the average and standard deviation of the replicates. And that will tell you how much variation you're getting within a lot. You do the same thing from lot to lot. And one of the things you can do here, for instance, is to say you run 3 lots where you're looking at within lot variation. But the averages you get here are also the same as lot-to-lot variations, right? You did 3 lots where you got 3 different averages here. And now there is a technique where you can then combine the lot-to-lot variations together, the standard deviations for the 3 lots, and you can average the 3 averages to get a lot-to-lot variation. Now, again, your statistician can tell you how to do this. I would say that when you're looking at lot-to-lot variation, you need as many lots as you calculated for individual units within a lot. In other words, you want to know your lot-to-lot variation at the same level of confidence that you want to know your intra-lot variation. So that's what I would suggest.
All right. Thank you, Steve. And one last question, Steve. We have time for one last question. And the question that's on my screen, which I have sent to you via chat messenger, the question is, you mentioned that there are 2 methods for specification setting, confidence intervals and tolerance intervals. Would you contrast the 2 methods and explain why you prefer the tolerance method?
Okay, the confidence interval comes out of the t-test. And basically what you do there is you calculate an interval around the average. When you do your confidence interval, you take a batch of replicates that you obtained, and in the case of setting specs for lots, you would take lot-to-lot averages. And from those numbers, you would calculate a confidence interval by taking the average and the standard deviations. Now remember, a standard deviation here is divided by the square root of n, n being the number of replicates that you put into the average. This gives you what is known as the standard error of the mean. Now, this is then massaged around and converted into a confidence interval. And what you get is an answer that says, okay, we have this mean and we have an interval which is based on the standard error of the mean. And we have 95% confidence that the future test results, now let me back up here. When you calculate the confidence interval, you multiply the standard error of the mean times a t-value, which you get out of tables for the t-test. And for instance, you go into the t table and you say, well, okay, I need 95% confidence. So you go and look in a t-table at 0.05 alpha, which is known as your chance of making a type 1 error. You want 95% confidence. You allow 5% uncertainty. You get that t-value and you multiply it by the standard error of the mean. This gives you a 95% confidence interval. And you're saying that I have 95% confidence that the true average, now remember that this is the mu, or the true average of all of these lots, is within this interval. And so basically what you're doing is you're defining the ballpark. You're saying we have 95% confidence that the true value lies in this ballpark, and you have defined the limits of the size of the ballpark. In a tolerance interval, you make a slightly different statement. And as I indicated, I think, to one of the questions I had, we look at this thing and we say, okay, we have this average. And based on this average, and you will find a constant. And this constant is multiplied times the standard deviation. So now the constant is designed, and it consists of many other constants actually. It's designed to allow you to make this statement, because you have to look this up in a table. And there are different constants. So you look it up and you say, well, okay, I want 95% confidence that 99% of the future test results are going to fall within this interval. Now notice I didn't say the true value. I just said that the future results are going to fall within this interval. So there's no statement about what the true value is for the average. We're just saying that in the future, 99% of the test results will fall within this interval, and we want to have 95% confidence that that will happen. And personally, this is why I prefer the tolerance interval, because we're basically admitting that, okay, future values, there may be some that will fall outside of that range. And, and we're saying that we want a certain level of confidence that that will happen. And that's what the tolerance interval gives you. Now, there is another thing. It's called a prediction interval. And a lot of people use the prediction interval for early development work. And what the prediction interval allows you to do is, for instance, you make 4 runs. And based on the result from those 4 runs, you can make the statement that we have 95% confidence that 99% of the next n, and n, say, would be 10, the next 10 lots will fall within this interval. Okay, it's the same statement as a tolerance interval, but you specify the number of results that are going to fall within that interval. And that gives you a prediction interval. Excuse me. But what this allows you to do when you're doing development work, excuse me, is to say, okay, we have 5 test results. We're going to do 5 more runs. And we predict that the future 5 runs are going to fall within this interval with 95% confidence that 99% will be in this interval. So you run 5 more. Now you got 10 previous runs, and you start to go from there. And usually what happens is that if you use prediction intervals, they eventually evolve into tolerance intervals, because you will have the numbers to set your tolerance interval. But basically, this is why I prefer the tolerance interval. It doesn't say anything about where the true value is or where the ballpark is. It just makes you predictions on future results. Now, the whole catch to all of this is that it's totally worthless if your process is not in a state of control. Because if your process is not in a state of control, none of these intervals work. And I'll put that out as a warning.
All right, thank you, Steve. Ladies and gentlemen, we've just run out of time, but I do appreciate all of you all with the questions and for Steve for answering them. Ladies and gentlemen, if you could look at the screens, you have the feedback form open on your participants panel. Please do go ahead and share your feedback. It's got about 8 questions. They are multiple choice in nature and would take you just 2 minutes of time to answer. We would really appreciate if you could fill up the feedback form. The reason is it would let us know if there are any positives and if there are any negatives which we can improve on. It's for the Global Compliance Panel team and for the presenter, Steve, as well. So if there are any feedbacks, we would definitely appreciate if you could fill up the feedback form. And also, if you have any further questions, like Steve said earlier, you could always send me an email, and I'll have that forwarded to Steve so that he can answer your questions. And also on your screens, while you fill up the feedback form, you could look at the upcoming webinars for Steve. And in case any of you are interested, you could always give us a call on our numbers or log on to our online portal as well. Our numbers are on your screens. It's 1-800-447-9407. Or you could always send in your suggestions or questions or comments to webinars@globalcompliance.com. And you have our email address as well here. And also on screen, once again, I'll just put forward the upcoming webinars for Steve. So in case any of you are interested, you could definitely put that forward. And ladies and gentlemen, just before we leave, Steve, would you like to give any concluding remarks for today's audience?
No, I think that is it. I regret the fact that, well, I shouldn't regret it. It's a short webinar, and we can't get into a lot of the details of the actual statistical calculations that you need. But I encourage you, take some training courses. And especially if you buy a software package, make sure you take the training course that goes with it. Thank you.
All right. Ladies and gentlemen, we'll end today's session. And I really appreciate all of you participating in today's webinar. And on behalf of the Global Compliance Panel team and the presenter, Mr. Stephen Kuhara, I thank you all for participating and wish you all a lovely day ahead. Thank you so much.
Okay, Michael. Thank you very much. I will sign off.
Thank you, Steve. Absolutely. Yep. Thank you so much.
Okay.
You take care. Bye-bye.
Bye.